Right, so the next speaker is Sam Lasham, who is a master's student here in the Department of Botany, and he's been looking at using environmental DNA and herbarium specimens to look at the diversity of Cordonerius. So I will hand it over to you.
Awesome, thank you, David. So yeah, as David just said, so this is my master's project, looking at the use of specimen and environmental DNA ITS barcodes to map Cordonerius species distribution and to determine their threat status. I've been doing this in collaboration with David Orlovich, Matt Larkham, Andy Nilsson. Obviously, you've heard from Andy just before, and Laura Van Galen, who you'll be hearing from after lunch, all of us from the Department of Botany at the University of Otago. So our ancient southern beech forests are a pretty quintessential New Zealand ecosystem, forming large forests both to the northern and southern parts of the South Island of New Zealand, with a bit of a beech gap through the middle. These trees, as some of these talks that I've previously discussed, obviously form symbiotic relationships with ectomycorrhizal fungi, trading important nutrients such as nitrogen for photosynthesized carbohydrates, and allowing these trees to thrive essentially. So traditionally, our southern beech trees have a pretty poor ability to expand their range past the historic forest margins. And this is sort of being hypothesized to be due to the absence of these pretty important fungal symbionts. One of the most prominent genera of ectomycorrhizal fungi that forms relationships with our beech forests is Cordonerius. Cordonerius genus is the most species-rich genus of gilled mushroom-forming fungi, with over 3,000 species described globally and approximately 250 described from New Zealand. Currently, it's sort of predicted that we've only identified around a quarter of the fungi in New Zealand, but we don't have an exact estimation of how many species might be existing specifically for the Cordonerius genus. And because we've only described so few of the fungal species, we currently have almost no data on the prevalence and ranges of these different species, which is obviously pretty essential information for determining whether some of these species are really common, whether they might be endangered and in need of protecting, or whether they play highly important roles in our beech forests. So I sort of started this project with two main research questions. The first one was trying to work out just how many species likely occur in the South Island of New Zealand and whereabouts, which regions they're existing. Is there sort of regional diversity in the species richness of Cordonerius? And the second was whether we could combine prevalence data from environmental DNA along with geospatial data to sort of look at the ranges of these species and work out whether they're quite prevalent or rare. So I got environmental DNA ITS sequences from multiple projects that had all been done in this department. This gave me a huge list of thousands of sequences. Obviously, these applicant sequence variants, as they're called, don't represent, each sequence doesn't exactly represent an individual species or even an individual, even just an individual member of a species. ASVs can also be quite variable from the ITS region within a single individual. So I needed a way to shrink these down into much smaller units, so that I could sort of work on a sort of pseudo species sort of level. So what I did is I grouped them into operational taxonomic units and tried to sort of make those OTU groups as similar to the groupings we traditionally see with species. And that was sort of going to be my way of estimating the amount of species richness in the South Island. And then, of course, had GPS data for a lot of these sequences, so I could use that to map regional differences and species ranges and that sort of thing. The environmental DNA data came from 3 different projects. Primarily, the majority of the data came from Laura Van Galen's PhD project on southern beech restoration and mycorrhizal ecology. as I said, you'll hear from her next. But that basically, she went out and sampled 81 different sites, so 810 samples, 10 samples from each site across the South Island, covering a large majority of our beech forest. A truly tremendous effort there. The second data set came on a paper published by Andy Neeson and Nelson et al. A couple of years ago on fungal diversity in canopy soil of Nophofagus menziesii. So that essentially they compared the fungal communities from the canopy soil with those of the terrestrial soil at the bottom. That was done sort of the southern portion of the west coast. And the third was from Marissa Straussine's master's project on trying to work out if Manuka leptospermum can facilitate southern beech colonization. So as you can see here, we've got sample sites from all across the island. This is almost all from Laura's work. The samples from that paper on canopy soil, they were taken from around here, and we don't have any geospatial data for the stuff from Marissa's project. Essentially, soil was sort of used and inoculation assays were done in Dunedin, so it moved around a bit, and I didn't want to sort of make any jumps that could actually just end up being cross-contamination from it coming from a different place. So yeah. So in terms of the pipeline I used, I got all these different projects which had used, between the three projects, had used two different sets of primers. So I trimmed the primers from the sequences, merged them all together separately, then filtered out everything with low read quality and that sort of thing. And then I needed to get all these ASVs into OTUs, as I previously discussed. So the most simplistic way to do that typically is just to do standard clustering of sequence identity. So you find some sort of minimum threshold that you cluster all the sequences by, and that's how you bring them down. Now with Cortinarius, typically these sequence thresholds are chosen quite arbitrarily. They're like arbitrarily, and I wasn't exactly very happy with just choosing an arbitrary number for this. So I thought, okay, well, let's work out what is a good cutoff to use for Cortinarius. So I went through and downloaded every Cortinarius sequence, ITS sequence off of GeneBank, split those up into every single species that was represented there, and then produced an alignment for every single one and worked out, okay, well, within each species, what is the sort of average shared sequence identity and the minimum shared identity, and averaged that out and found that, okay, well, for sequences on GeneBank, on average, they share no less than 97% of their, no less than 97% identical ITS sequences, and on average, they're usually about 99% similar. Obviously, GeneBank has heaps of issues with misidentified specimens and that sort of thing. So these numbers are probably lower than the actual truth. So just to sort of, as a more in-depth, more rigorous study method, I did a similar thing, just looking at 15 species using sequences from specimens from the department here at the University of Otago. These had all been looked at quite in-depthly phylogenetically. So I could be a lot more sure that these species were what we thought. And I did the exact same thing and found that, you know, on average, about 99% was sort of the minimum cutoff for those. And on average, they shared just shy of 100% actually of their sequences. So that sort of led me to go, okay, well, 90% is a pretty good cutoff for clustering things into a sort of species level of OTUs. Then ran through an algorithm which is sort of set up to remove things that might be errors from sequencing, or they might just be highly similar sequences that are definitely from the same group. I then got these and used BLAST, find a whole bunch of similar sequences, name sequences, and produced a huge, enormous tree, which I could then look through, and I could use the tree and BLAST and sequence similarity to assign species to the OTUs that I'd created, and also to look for new species, undescribed species. So the main point of the main first research question was to look at the species richness of Cortinarius. We identified 588 OTUs, which were identified as Cortinarius, representing over 400 clades. 121 of these were able to be assigned to 96 species. So there's approximately 250 New Zealand species, and about 200 of those have been sequenced. So essentially in this project, half of the species that are currently known that could have been found were found. So that's interesting to look at. But on the flip side of that, 467 OTUs forming over 300 clades couldn't be assigned to a species. So approximately 3/4 of all the things that were discovered in these data sets are currently undescribed, which shows that, hey, we've got a good bit of work to do in terms of really characterizing this genus. And that's quite consistent with that paper at the start that reckoned about only 1/4 of the species in New Zealand have been described, well, fungal species in New Zealand have been described. So I produced some species accumulation curves just to see if we'd sampled enough of the South Island to have a good estimation of how many species we've found. So as you can see, what we want is this curve to sort of get close to a plateau. The more samples we get, the more things you tend to find. But after a while, once you've found everything, no matter how much more you sample it, obviously you're not going to be finding anything more. So you get a plateau. So we can see here we've got a pretty good accurate level of sampling for working out what is present in the South Island. And if we extrapolate this relationship out to a plateau, we found that, okay, there's around 640 OTUs in the South Island of New Zealand. When the regions are viewed individually, however, it appears that quite a few of them definitely need to be looked into more to find other species there. Nelson being a prime example here, there's hardly even a curve. That's because there was just a single site in the Nelson region. Only 22 OTUs were found, clearly probably due to the low sampling of that area. And on the flip side, Mount Aspiring was a much more highly sampled area, 14 sites, which is 140 samples, and highest number of OTUs of any region that was studied, so 220. So this is getting a lot closer to plateau. Obviously not as good as the whole of the South Island, but looking much better. So I mapped these projected, the projected species richness across the South Island. And you can see here I've used an asterisk to sort of show the ecological regions which are reasonably well sampled, and most of the highly forested areas are. Only areas worthy of note that probably need some more sample and have a lot of beech forest sort of around this area and around Fiordland as well. Interesting sort of Nelson, as I showed before, obviously quite low number of OTUs, quite low level of sampling, and wasn't very well sampled. Whataroa was similar to Nelson in terms of the only having one site. However, due to the fact that it has a significantly reduced level of beech forest, primarily because it spans this so-called beech gap across here where there is very little beech forest, we actually did sort of get a pretty decent species accumulation curve for that. So that showed that region actually does just not have very much in it, as would be expected due to the limited amount of forest. So sort of moving on to prevalence and threat status. So sort of non-lichenous fungi are currently the only group of organisms in New Zealand which haven't actually had their conservation status assessed by the Department of Conservation. which is obviously highly problematic being such a hugely important kingdom. Largely this is just due to the large number of data deficient fungal species and difficulty of quantifying fungi populations. We can't go out and count how many fungi individuals exist in an area very well. And as Lydia touched on sort of in her talk before, so that's obviously a huge problem. We need to work out a way of quantifying how prevalent these things are. So I sort of went, well, okay, what's the easiest way to do this with all the data I've got? Let's just list the things by the proportion of samples that they're found at. And what came up was essentially 149 species that are known to exist in New Zealand were not detected in this study at all. Around 50 of those haven't even been ITS sequenced. So that explains why they couldn't be detected. They might have been actually detected, and they might be lost in some of those OTUs we've got, might actually be representative of those species, but we have no way to know until those are sequenced. And also, but that still leaves about 100 sequences that weren't found versus just under 100 that were. So we can't really confidently conclude that just because something wasn't found in the study, it is rare, as obviously there were so many things that were not detected. And also some things such as Cortinarius epiphyseae, which, you know, there's 40 collections of it. So I assume it's not super duper rare, but we didn't find it whatsoever. So that sort of is that. So some examples of some of the species that are quite prominent. Cortinarius across was found in 14% of our DNA, eDNA samples, and as you can see here, this species is known to exist across the South Island, and this sort of just proves that this shows quite well. If you can see, as I've mapped across here, these green dots representing all the sample sites that we found the species at, well, a sequence that related to the sequence at the species at, and the triangles representing collections. You can see that it's sort of found all over the place and clearly pretty prevalent. Now, moving on to a similar thing is Cortinarius vitriopiliatus. Now, this is listed by the Department of Conservation as a data deficient species, which essentially means they just don't have enough data to work out, is this highly prevalent? Is this something that's endangered in need of protecting, something like that? So this OTU that sort of looked like it was Cortinarius vitriopiliatus was found in 16% of the samples across the South Island. There's also about 25 different collections found. I should note that not all the collections are actually shown here because I couldn't get GPS coordinates for all of them. But, you know, this shows it's pretty prevalent, and that's looks to me at least that there's some pretty good evidence there that we can start looking at that data deficient status and hopefully making some moves to get that changed. Another similar example is Cortinarius arnauensis. This is slightly different in the fact that I could only find evidence of 3 collections of it. Never been observed on iNaturalist, or, well, that probably because it's quite a rare, not very well known species. But we did find it in 12 sites from quite a few different places. 3% of the samples, so considering half the species sort of known to exist weren't detected, that's reasonably prevalent, all things considered. So that's another data deficient species that I think we can probably start to look at updating the status of. Now, on another little side note here, here's something which, based on the data we have for the things we identified, obviously looks quite rare. It was only found in 1 site, and actually only found in data from that Canopy paper. It was found in 18 different samples, and this OTU looked very similar to an ITS sequence from the holotype of Cortinarius comptulus, but it's also very more than 99% similar to Cortinarius hemitrichus and Cortinarius jacobii. All 3 of these species are only ever found from the northern hemisphere, not found down south whatsoever. So that's really interesting that we found that actually. And one thing I did manage to find is there's one collection of hemitrichus that Jerry Cooper found in mid Canterbury, I think around here somewhere. So yeah, so maybe that's what this is. It's pretty much impossible to tell without going there and trying to find a specimen and just crossing your fingers and hoping that it pops there up on the day that you're there. So, you know, who knows what that is, but that's something that might be interesting for further study, especially considering, I think, in that paper, there was an introduced hepaloma found as well that wasn't known to be from New Zealand. So maybe they're coming from somewhere. Not sure. That'd be really interesting to look into. So that's a good example of, you know, looking at the species we know exist and going through and showing how prevalent they are. But obviously, as I said before, the vast majority of things we found were unidentified. The most prevalent of these was OTU 105. As you can see, highly prevalent, found all over the place, all over the island in eDNA, and you know, no idea what this thing is, but you'd think something that prevalent would have been found quite a few times. A specimen would have been sequenced, we'd have worked that out. So had quite was kind of similar to Quaternaria leptospermorum, but once I sort of looked phylogenetically, maybe not so much. I sort of blasted it out, and there's a few sort of unidentified specimens from Tasmania which were reasonably similar, and then obviously our Quaternaria leptospermorum, which is similar as well, but you know, nothing that's super evident. So I made a big phylogeny anyway, and OTU 105, you can sort of see at the top, there's not great branch support here, but one thing that is quite clear is it doesn't really seem to be grouping with very closely to any species in particular. Quaternaria leptospermorum, have you noticed before, these are the two sequences that were most similar, which seem a bit distant, and the other one is sort of down here. So quite interesting that it showed them separate. I mean, the branch support values for separating these groups are pretty low. So yeah, so that's quite so, you know, maybe they are the same. This probably needs to be looked into a bit more in depth. I think the next plan is to sort of branch some of these OTUs out and find the ASVs that were grouped into them, because by having more ASVs on a clade, it sort of increases some support of actually helping the program separate this out and create a better phylogeny. So OTU 339 was another reasonably prevalent species, found in 7% of the samples, seen throughout South Island. 13th most common thing, found all up. So pretty common, as you can see here. It just sort of grouped up with a whole bunch of other OTUs. The only thing even remotely close was Quaternaria swiporianus. So once again, produced a phylogeny, and it did actually group quite closely to a sequence of Quaternaria swiporianus, although with pretty low branch support. Which, interestingly, the sequence it came out quite close to is always separated. No matter how many times we did it, there's another phylogeny I'll show you next, but always separated from this larger group. And there's quite a lot of OTUs involved here. So I'm unsure if this is misidentified and it's separate from these, or what seems more likely is maybe that these are all Quaternaria swiporianus, and that is just a group that is much more diverse in its ITS regions than a lot of other species in Quaternarius, and therefore, because it's more diverse and we sort of used an average cutoff for producing these OTUs, quite a lot of OTUs are actually included into that group. So that's sort of something that needs to be looked at, and we did what I mentioned before. So we did add in ASVs into this rather than OTUs, and we got a very similar thing, although because the ASVs were show a bit more diversity than the grouped up 8 OTUs, we get much better branch support values for sort of separating these two groups here and here. So yeah, there's definitely something going on there that needs to be looked at, but that's sort of work in progress at the moment. So anyway, in summary, at least 630 to 650 Quaternary species are predicted to exist in the South Island of New Zealand. Further sampling is definitely going to be required to improve the accuracy of that prediction, particularly just in those regions I noticed that might be hiding some interesting things. I mean, Fiordland and Inland Marlborough were missed out a bit. Approximately 3/4 of the OTUs discovered in the study could not be identified as any known described in ITS sequence species. So so we've only actually found about 1/4, which is sort of consistent with that other study. This research has been pretty useful in determining a number of Quaternary species as highly prevalent, but a larger sampling effort is probably going to be required to confirm any species as rare or endangered with confidence. And just as a sort of side note, the Northern Hemisphere species, sort of maybe Seconchulus or Quaternarius hemitrichus, may be present in New Zealand, but a specimen collection is going to be necessary to determine whether the species has been detected or whether a New Zealand species might exist with the same or similar ITS sequence. Cool, thank you. Any questions?
Great, thanks, Sam. So I can see David Hera's got a question in there. Maybe we can get you to ask it in real life. It's more realistic when you talk, isn't it? There we go. You should be able to do that now, David. Do you want to stop sharing your screen? You can stop sharing your screen, Sam. Then I don't have to look at myself delayed.
Sorry.
Good.
Okay, yeah, I was just curious about looking at other loci as well as ITS2 for maybe helping to further resolve quaternarius, because with Pleurotus, for example, there's a lot of literature supporting that ITS2 is not informative enough when the species are closely related. And I wonder if that might also be the case there. So, for example, I'm doing RPV2 and a translation of elongation factor alpha as well as ITS2 for multi-locus phylogeny.
Yeah, exactly, and that's a huge thing. Obviously, basically, I've done any field work for this. All the research projects were they were already done, and I just came along and grabbed the data, and none of them were done with this sort of project in mind. This came along later, so very that obviously would definitely improve the accuracy and make it easier to split a lot of these things out, because there are a lot of groups that are hard to split up just with ITS. But yeah, obviously that wasn't done initially, so we couldn't do it. But I think just the eDNA sequencing, particularly with the large number of sequences that Laura was doing, was reasonably expensive. So to do that for two genes might also make that quite expensive. But yeah.
Right. Yep. I think you've got to be careful not to throw the baby out with the bathwater, though, because a lot of the phylogenies that have included ITS and other markers of, in, say, Quaternarius, for example, are largely concordant. So I don't know that it needs to. And the other issue with environmental DNA is that if you had a different marker, you couldn't necessarily match each OTU or each clade or whatever up with the equivalent one in ITS, unless you had your reference species as well. That's fine. Okay.
Yeah, I think it would be a lot more work, maybe, and a lot more to probably only sort of resolve a very small number of species, maybe.
Yep. Any other questions there? Oh, yep. Thanks for that. That's good, David. One of the things that, if we're waiting for anyone else to think of a question, one of the things to mention is that both the hebeloma that was found, the introduced hebeloma that was found in that Haast site, and the Quaternarius that you mentioned, that where there was a collection from Jerry Cooper from Canterbury somewhere, both of those are known to occur elsewhere in New Zealand. So that's perhaps not so surprising that they're in New Zealand, but it's more so surprising that they have a host that is a native species rather than an introduced one. Okay. Peter J's just asked the data on numbers. Oh, right. Yes, the data on numbers of unnamed things are good to have. Absolutely. Yep. That's why it's so exciting, actually. And it matches up with your or various people quoting that number of 25%.
Yeah, no, exactly. That's really cool how concordant that is.
Something Sam's been working on is trying to, because, the OTUs are kind of the finest, well, the ASV sequences are the finest level, but then you can group them into OTUs. But then when you do a phylogeny of OTUs, not all of those clades of OTUs have very good branch support. And so that's the challenge, to sort of work out how to automate drawing the line between, to find groups that have reasonable phylogenetic support.
Yeah, you could clearly see those Cortinarius Wyporianus groups. The second you jump to ASVs, the actual structure of the tree doesn't change so much, but the branch support sort of jumps very, very much.
Yep. So Camille's just said similar number in Patagonia too. That's fantastic. And Andrew Leach has just said, sorry, there. Yes, these are being recorded. So these will be able to be shared as long as the speakers are happy to have that shared. Great. Anybody else? Oh, yep. Need to get the Australian, this is from Teresa, really need to get the Australian data for the Cortinarius truffles. Oh yeah, that's right. Yeah, you sure do. That's a good plan. So something that Sam mentioned as well is that, he's found things that have a genetic similarity to Tasmania, and that's something we constantly, every time we blast something, every second time we blast something, it comes up with one of Bryony Horton's clones from her PhD, was it? Yeah, in Tasmania. So, yeah, I reckon we need to get Genevieve to host us over there to get a good lot of DNA sequencing and a fungal foray.
It's very interesting. A lot of it seems Tasmania far more than other parts of Australia as well. I mean, obviously similar climate, but
yep. But we don't know if that's a sampling bias or not. But yeah, it sure is. Yeah, that's right. Good. Excellent. Okay. Anyone? Oh, what do we got here, Teresa? Common pattern. Look at the Lactifluus Clarkia group paper. Yep. 10 North. Oh, right. North Island is more overlap with Queensland. There you go. Cool.
Interesting.
Right. Okay. Well, if that's it, then I think we can probably call that. Oh, here we go, Peter. Peter Johnson has asked any feel if there is any difference between the Nothofagus subgenera. So I don't think Sam will be able to answer that, but Laura might be able to in her talk after lunch. So you might want to hold, unless you want to say something, Sam. But
I haven't looked into that.
Yep. Oh, and Laura's given a thumbs up. Yep. Laura's.
Yeah, we'll leave that to her.
Awesome. Yeah. So that's a great little segue into the data some of the data that Sam was collected by Laura Van Galen for her PhD, and she has done a wonderful project that there's a lot to talk about there, and she is going to talk about that at 1:30. So I think that'll be something to look forward to, and that will she will answer your question there, Peter. Okay. Great.
Cool.
All right.
Awesome.
Well, I'm going to say thank you, Sam.